Good morning students, welcome back to my channel Molecular Mysteries. So in the last class we have started a new topic about cancer. So we discussed the terms, basic terminology in cancer. So in this class let's talk about the properties of cancer. So we have gone through the thumbnail. So this class is going to be a really big topic because it's not easy for you to understand. So I'm also giving you the basic diagram for every property of cancer, how it is different from the normal cell. Okay, so without any further delay, let's get into the class. So today let's talk about what are the properties of cancer. So the main important thing is that for a normal cell, it has to undergo a proliferation, it has to undergo a differentiation and the survival. These are the main important terms for a normal cell to exhibit. It has to undergo the basic property of undergoing proliferation, differentiation into other cells and the survival capacity. So when you differentiate this particular property of normal cell, the cancer cell doesn't have a proper cell signaling mechanism, due to which there is no proper proliferation inhibition. It doesn't have proper differentiation. I can say it is an abnormal differentiation, and I can say the survival capacity also. Okay, now let's talk about what are the main properties of the cancer cell. So if you talk about the properties of cancer cell, the first topic is, right, these are the properties. Let's go in detail about the properties. First one is the density dependent inhibition of cell proliferation. So what does that mean? Like each and every normal cell has got a cell proliferation capacity based on the nutrients that you supply. It's like if you take a petri dish having a growth serum medium, added normal fibroblast cells or any endothelial cells or epithelial cells to that, to which based on the presence of the growth factors, these generally grow and differentiate based on the cell density. Like each and every cell, a normal cell has to normal cell has to reach a normal cell density, upon which they stop the proliferation and enter into a G0 phase where the cell cycle is arrested. That is what you call a quiescent phase, right? At that phase there is no further DNA activity or DNA synthesis happening. I don't say it's a cell death mechanism, but the growth of the cell is being maintained, or I can say it is cell proliferation is completely inhibited, right? This happens in a normal cell, but for a tumor cell, if you go on under a petri dish, they keep on multiplying. They doesn't follow the cell signaling mechanisms that normal cell do. That is what the differentiation happens. That is what the cell proliferation is completely inhibited. No matter the growth factors are there or not in the medium, these tumor cells or cancer cells keeps on proliferating and increasing their number. So this is called the cell inhibition, density dependent inhibition of proliferation. This particular property of normal cell is completely absent in the tumor cells. This is the main important property of the cancer cell. The second one is the autocrine growth stimulation. What does this mean? So you all know that a cell needs growth factors for undergoing many mechanisms, right? If you are growing a cell in a culture, you need to supply that with basic growth factors required. So based upon the growth factors, like if you take the fibroblast cells and you are growing them in a culture, so based upon the growth factors present in the serum, they keeps on proliferating and multiplying, right? They are completely directly proportional to the amount of growth factors to the amount of cell proliferation. But what happens here, the tumor cells, they are not dependent on the growth factors that we provide, because tumor cells have the capacity of producing their own growth factors. Okay, they can auto stimulate themselves. That is what you call autocrine stimulation. Means the growth factors that are required for the tumor cells are much more than the growth factors that we supply from outside, because the tumor cells need the blood supply. The tumor cells need more nutrition for increased proliferation. All these things are produced by the self, by the own cells. That is what you call autocrine growth. All the cells produce their own growth factors. All the tumor cells produce their own growth factors, which are required for your tumor to develop, or to tumor to multiply, or even for the metastasis to happen, right? And the third important property of the cancer cell is loss of E-cadherin adhesion molecule. What is that? Each and every cell is having E-cadherin. This is called the adhesion molecule. This molecule is required for the attachment of this particular normal cell to the other cell. That is how the cell to cell interactions happen. That is how the cell to matrix interactions generally happen. right? But for the tumor cells, this particular E-cadherin adhesion molecule is completely absent, which makes it different from the other normal cells. So generally, you can say that tumor cells are little rounder in shape, and it doesn't have any interaction with the cell, and the same time it doesn't have any interaction with the matrix, right? That is how it makes it different from the other normal cells. At the same time, this has the capacity, the tumor cells without the without the E-cadherin molecule has a capacity to enter itself into the connective tissue, like it can enter even into the blood. That is how it is. Metastasis is also one of the important thing for a cancer cell due to the loss of this particular E-cadherin adhesion molecule, right? So the other important, I told you, contact inhibition. So I told you cell to cell contact is very important for cell signaling pathways, so that they maintain an array of cells. So if you see, for example, if you are just growing a group of normal cells in a serum and you are observing it under a microscope, so whenever the fibroblast cells, I'm taking it as an example, so when these molecules, they keep migrating, once they have a touch with the neighboring cells, they stop migrating and they keep on multiplying and having a neat array of cells. You can see under the microscope, neat array of cells are formed when you normally see a cell multiplying in serum. But when you see a tumor cell, this particular tumor cell, though it attaches to the neighboring cells, it doesn't leave the capacity of proliferating, right? So it keeps on multiplying in all the other directions without having a proper cell to cell mechanism or cell to cell interaction. So this is also one of the important properties of your cancer cell. And I told you last one is the secretion of proteases and collagenases. So what does that mean? So I told you proteases and collagenases. So for proteases which act upon the protein, right? Proteases are the enzymes that act upon protein. Collagenase is an enzyme that acts upon collagen. So when these proteases are produced by the own cancer cell, the whole cellular matrix, extracellular matrix components are completely digested by these proteases, which makes it easy to enter into the normal healthy tissue cells. Hope I am clear with the concept. Proteases are like, they are degrading, they are digesting the basic extracellular components, thus it is easily invading to the nearby neighboring tissue cells which are healthy and normal. So that is how the metastasis starts, like it starts damaging the other tissue cells which are healthy. In the same way, when you talk about the collagenase, it actually enters into the connective tissue, right? It damages extracellular protein and it also enters into the connective tissue. Thus it is easily, the cancer cell is acquiring that particular capacity of metastasis. So these are the basic properties, and you have also, I will also show you one important diagram for you to understand. Okay, right. So I told you this is the main density dependent inhibition. Normal cells generally proliferate and cells become quiescent state. So what does quiescent state means? It doesn't enter into any cell further cell proliferation. So the cell cycle is completely arrested and have the reduced metabolic activity, and they're not dead, right? They just have the reduced DNA synthesis. This generally happens in the normal cell culture. When you take the tumor cell culture, when the cell proliferation starts, no matter how amount of growth factors that have been supplying into the serum, they keeps on continuously growing, and thus the cell proliferation is continuous in the tumor cells. For your understanding, I am just giving you a diagram. Now let's talk about other important factor. I told you autocrine growth stimulation. So these are the autocrine, the tumor cells which have the capacity of producing their own growth factors, and thus see they are increasing its number. That is how it keeps on multiplying. Each and every tumor cell has a capacity of producing their own growth factors. Thus it also helps in angiogenesis. In the last class we discussed about what is angiogenesis, right? Angiogenesis is the capacity of making the cell to get vascularized, like it helps in blood supply. So when it takes a bypass from the main blood vessel, and thus the angiogenesis produce their own blood vessels from the main capillaries. So from those capillaries it starts producing the nutrients. It helps in giving a good supply for the tumor cells, because it needs more and more blood supply, because it's having more and more number of cells. They all need to have the nutrition and have the cell proliferation capacity for the cancer cell, right? So that is how the autocrine growth stimulation is also one of the important factors for your cancer cell. And I told you proteases and collagenases. So this is a small adenoma that is produced here, that is grown here. See, I can explain it better. One second. Yes. So this is a small adenoma that is growing here, right? So because of the enzymes called as proteases and collagenases, what happens finally, that tumor is becoming a malignant tumor because it is invading into your connective tissue, and finally it is invading into your blood and lymphatic vessels also. That is how a normal tumor is becoming a malignant tumor because of the enzymes called proteases and collagenases. Okay, now let's go for the third diagram. Yes, this is what you call defective differentiation. So for that I will talk about the theory part, then I will talk about, right, these properties are done, right? So let's go for some more properties of cancer cells that differentiates that from your normal cells. Okay, hope I am not confusing with the topic, because these are the main important properties of your cancer cells. Then they might ask you any in some or the other way. Okay, now let's see what are other important properties of the cancer cells, right? I told you it is a defective differentiation. What is defective differentiation? So defective differentiation means they fail to differentiate. So till now we are talking about the cell proliferation capacity and all. And what is defective differentiation? See what happens, you all know we have pluripotent cells, or you have the stem cells, right, which come from the bone marrow, right? You have group of cells where the cells get differentiated into granulocytes, monoblasts, right? These are all the different type of cells that you get, the lineages, myeloid lineage, right? You get reticulocyte, you get erythrocytes, RBC, right? Granulocytes, everything you get under the defective differentiation of cells. But when it comes to the cancer cell, that defect will happen. For example, if you take the leukemia, which is called the blood cancer, so the patient with leukemia have the highest number of WBC in his blood. Why? Because they are all having the defective differentiation. They should actually get differentiated into different type of body cells, like, but instead of that, what happens, the leukemia keeps on undergoing more and more differentiation without any cell apoptosis, right? Well, you know that apoptosis is a programmed cell death, which really happen in normal cells, right? So these normal cells undergo a programmed fashion of cell death, so that that phase of cell is completed. But for the tumor cells, due to the lacking of that particular capacity to undergo apoptosis, they undergo defective differentiation, so which leads to increase in the number of white blood cells without getting differentiated. Hope I am clear with the concept, right? So I told you here it comes to fail to undergo the apoptosis. This is important, due to which this defective differentiation happens. And I told you angiogenesis is also one more important property of cancer cell, which have the capacity to produce new blood vessels from the capillaries, and thus giving more and more blood supply to the growing tumor. The main important condition for a tumor to develop is its number. For that, it needs more and more blood supply. For that, they start having the angiogenesis, which takes the blood from the normal blood flow, and due to which these cells keeps on increasing as they get nutrients through the blood, right? And fourth one important thing is, let me complete this, then I will talk about what is defective differentiation. Yes, so I told these are the pluripotent stem cells. They should have normally undergo, this is a normal condition. Okay, this is the normal condition that normally happens in our individuals, right? So this is a normal cell division where the pluripotent stem cells undergo a differentiation, forming finally RBC. So I'm only talking about the RBC. So I'm giving you an example as RBC. So you have a correct differentiation of your stem cells, but defective differentiation happens in the tumor cells, where this WBC keeps on increasing its number. So that is why I told you leukemic cells fail to undergo the differentiation, thus leading to the continuation of your division, increasing in the tumor cells. Okay, so this is the topic that I want to talk about in this class. Yeah, sorry, I thought of discussing about the expression of telomerase. Right? What is expression of telomerase? So you all know telomeres are the ends of your chromosome, right? So when I talk about the apoptosis, generally the cell death happens. Even the cell's DNA is also completely damaged, and you have no further proliferation of the cell. But when what is actually telomeres? Telomeres are the ends of chromosome. For example, if you take this as a chromosome, the ends of the chromosome are generally protected by telomeres. They are the ends of the chromosome, right? Like a cap-like structure. So what happens? The cancer cell, what actually it does is it has to make the DNA damaged. You all know that the DNA is actually damaged in the cancer cell. So how it does happen? This particular cancer cell, first of all, starts releasing an enzyme called telomerase. This telomerase mainly acts upon the ends of the chromosome. If you take this as an example, if the ends of the chromosome are mainly affected by the telomerase which act on telomers, the DNA finally gets intact. It finally gets exposed, due to which the DNA is easily damaged by the cancer cell. So these are the main important properties of your cancer cell. Hope you are following this class thoroughly. This is the important property. If you are well aware of these topics only, then I can talk about proto-oncogenes and what are the types of viruses that attack the body and what are the types of cancers. Okay, in the next class I will talk about that, and if you have any doubt, please let me know in the comment box. Hope I am giving you a clear example or clear discussion about the properties. So if you have any doubt, please let me know. Please try to answer the questions that I've been posting in the community tab. So thank you and have a nice day. So see you in the next class with the other with other types of tumors, types of viruses that attack the cancer, like viruses that attack the normal cells producing the cancer. Okay, so thank you and have a nice day.